Okay, hi. So we're going to go through Module 4's video is for defense responses and plant resistance for your plant immunity fall 2020 class. So we've learned a lot about the different forms of resistance, constitutive resistance versus inducible resistance, but what actually produces resistance in the field? Well, the answer is a mixture of many different things, but sometimes things beyond what I've actually told you about. For example, a lot of pathogens produce toxins. So toxin sequestration or degradation is actually a major tool that we can use for producing resistance. And then there's also some artificially generated forms of resistance that I'll show you some examples from today. And then there's actually a lot that we just don't know. For example, there's forms of resistance that we've tracked through using breeding, but we don't understand actually how that resistance is generated in plants. So there's actually still a lot to discover. So here's an example of toxin degradation as a tool for generating resistance. So in this particular disease, we have Fusarium head blight. It infects wheat and barley and corn, and it produces a virulence factor called deoxynivalenol, and it's called DON. So DON is highly regulated because it's also causes this disease in animals. And so to make sure that our grain crops that we're trading doesn't have too much of this toxin, we really have to measure it in all of the shipments and all of the wheat and barley and corn that we produce. So this is quite an expensive problem. And these people track one form of resistance to this particular disease through looking at detoxification of DON to basically conjugated form that's no longer toxic to plants. And so it's no longer a virulence factor. And the interesting thing about this story is that the detoxification gene didn't actually come from plants. It's not present in plants normally. It came from this fungus that it's an epiphyte and is growing within and on top of the ancestral wheat that we used to actually generate and domesticated wheat. So this is a case of horizontal gene transfer for this particular detoxification gene. And that got lost originally when we domesticated wheat, but they were able to do like a wide cross and bring that gene in from the wild species of wheat. So we have a case of both horizontal gene transfer, which is quite rare between microbes and plants, but also wide cross, which generated this form of resistance, detoxification. We also have mechanisms for, of course, transferring receptors for MTI or ETIs. We've learned about them between species. So again, this requires just transgenics, basically cloning the genes out and then producing them in a background that they wouldn't normally exist in. So for example, this is the EFR MAMP-triggered immunity receptor for a bacterial MAMP. It was transferred from Arabidopsis into tomato, and you can see it's produced quite a lot of resistance to this particular bacterial pathogen, which is Ralstonia solanacearum. And actually, the interesting thing about this too is that if you transfer that EFR, you see more disease resistance, but you don't see any changes in rhizobial symbiosis in the roots. And so, again, not all MTI receptors are the same. They work sometimes with some pathogens, and they don't actually inhibit symbiosis, which is pretty interesting. We also have transfer, for example, here of an R gene. This is the BS2 R gene, which is transferred from pepper into tomato, and you can see that there's bacterial resistance here. So this is a successful strategy, but one thing that actually, or let's go back here. One thing you have to realize is that the machineries that are required for all of the signaling downstream of either the PTI or the MTI or the ETI has to be present in that host for the transfer to work. So this has also been a way that people have been trying to discover what are the differences and similarities between different plant species. And, you know, we can do this successfully between some types of species, but other species are actually kind of too different to successfully transfer receptors. Another interesting way of bioengineering resistance is by completely taking a module of the R protein with the decoy. So normally you remember that R genes will actually guard a host protein, and if modifications are made to the host protein, then they'll activate ETI. So you can take that sort of entire module, so 2 different genes, and then put them into a different host instead of relying on just transferring the R protein itself. And in this case, this happens to be a signaling module that's kind of well conserved. And so what these people did was they have this R protein here that's guarding PBS1 at the plasma membrane. AVR-PPHB is an effector protein that's a protease. It actually finds this little sequence of amino acids and cleaves at that specific site. And that cleavage will activate the RPS5. Now, the idea here was they actually just modified this little site depending on the protease that was present for a specific pathogen. So instead of relying on this avirulence gene being present to produce the R gene mediated ETI, they could actually modify this to whatever protease the fungus or the bacteria was producing, as long as they knew what the recognition site was. And so this is, I think, a really interesting way of adapting kind of a specific form of resistance to almost any system, as long as you know enough about your pathogen to know what kind of protease it is carrying. You can definitely use this to your advantage. And they did this successfully to generate resistance to viral pathogens, for example, bacterial pathogens, and I think they tried a fungus as well. One other major area that I should mention is susceptibility genes and targeting those types of genes. So these are genes that, again, when they're present in the dominant form or when they're transcribed, for example, they produce susceptibility. And we don't, sometimes we don't understand exactly why they're producing susceptibility. For the case of some of these genes, it just happens to be a gene that the pathogen needs in order to survive or proliferate in the plant cell. But this is one of the most famous susceptibility genes. This is the MLO gene, which is involved in powdery mildew susceptibility. So when you knock it out, or you have a recessive form of this gene, you see really nice resistance. So here you go, here's the resistance in tomato. The cool thing about this one is it's actually conserved in monocots and dicots, and it is one of the longest lasting forms of resistance, one of the stablest resistance we've ever seen in plant pathology. Also really interesting is that we don't know why it works. We don't understand how it really contributes to susceptibility, but we do know when you knock it out, you get spontaneous cell death and callus, so there is a bit of a yield cost to knocking out this gene. And I think this is going to be one of those big question marks that if someone figures it out would be like a big advance in our field. So I'm excited to see if someone can do that. So far, there's been some hints, but nothing conclusive. Another important susceptibility gene you should know about is the EIF4E. This is, it's hard to say, but it's basically involved in initiating translation. And this is absolutely necessary for polyviruses to replicate within the cell, because polyviruses are basically RNA viruses. They have this protein that allows them to bind to that particular factor, and this recruits the translation initiation. So for them to translate their RNA and start reproducing more of themselves, they need to have that interaction. And so people have found natural mutations in this gene, and that natural mutation has generated quite a lot of resistance. But because it's natural mutation, there's also been, sorry, not natural resistance, it's also generated a lot of like counter strategies by the pathogen. And because viruses in particular are very good at mutating quickly, they generate a lot of mutations through really interesting mechanisms I can't get into today. But viruses are able to overcome a lot because of this variation that they have. And so you see a lot of progression and evolution over time of changes of this particular gene and corresponding changes in the viral VPG gene so that they can overcome that type of resistance. So it's not stable, but it is still useful in some plants. Okay, so there's a whole lot of other examples that I don't have time to get into, but just realize that in general, the more you know about your system, your pathogen, and how it's actually overcoming your inducible responses, the better chances you have at designing something that will produce resistance. So while knowing the plant immune system is important, you also need to know how the pathogens are manipulating that immune system. And I think in the second half of the course, you'll see a lot more examples of, Allison will talk about how pathogens are actually able to overcome the immune system. And there's lots of really interesting strategies there. So for future prospects, I just wanted to mention that, again, CRISPR is a new tool for generating changes in the genome. And this has created a lot of excitement around our regulation of genetically modified crops, and so potential changing the way that we produce resistance in crops, because bioengineering resistance is not always easy if there's no public acceptance of using that type of tool to generate resistance. So with CRISPR, we're seeing not complete deregulation, but changes in regulation around how we view those types of crops. So we'll see how that changes in the future. But we also, through the power of genomics and these amazing tools that we have now to understand the variation in genes across species or within a species or with ancestors, now we can really design wide crosses, so crosses to an ancestor with a cultivated or domesticated crop much more easily. And so that should also generate some really interesting changes in the future. So with that, I will talk to you next time.